Imagine for a second that you are a single cell.
Just a tiny microscopic bag of fluid, basically.
Exactly. You don't have eyes, you don't have ears, and you certainly don't have a brain.
Right. None of the sensory organs we rely on.
But somehow, if someone squishes you into a tight space or stretches you out across a stiff surface, you just know what's happening.
Yeah, you sense that physical push or pull instantly.
And you immediately change your behavior to survive. You might start reading different parts of your DNA, or terrifyingly, you might even turn into a cancer cell.
It really is wild when you think about it.
It is. How does a blind, deaf bag of fluid actually feel a pinch?
Well, it's kind of the ultimate locked room mystery of biology, isn't it?
It totally is.
Because we're talking about a cell needing to convert a purely physical, mechanical force into, a chemical language.
Sure.
A physical squish has to somehow translate into a very specific command that the DNA inside the nucleus can actually understand.
And that translation process, that is the beating heart of today's deep dive.
Oh, absolutely.
We have a dense, incredibly groundbreaking paper in front of us from Nature Cell Biology. And our mission today is to unpack the intricate, hidden architecture inside our cells.
To reveal this newly discovered mechanical switch, yeah.
Right, a switch that controls everything from how our healthy tissues grow to how our bodies actually suppress tumors.
So to really grasp how this works, we should probably introduce the main characters of this microscopic drama.
Please do. Who are we looking at?
Inside your cells, you have these two executive proteins. They're called YAP and TAZ.
YAP and TAZ. Got it.
Yeah, and you can think of them as the co-CEOs of cell proliferation. When YAP and TAZ manage to get inside the cell's nucleus, that secure headquarters where all the DNA is kept, they just start shouting orders.
Like telling the cell to grow, multiply, and divide.
Exactly. They are permanently trying to hit the gas pedal on growth.
But the fundamental question has always been about control, right?
Yeah.
How does the physical shape of the cell actually tell these executives what to do?
Yeah, that's the big puzzle.
Okay, let's unpack this. Before we get into the new discoveries from this paper, we really need to understand the mechanism of how a cell feels anything at all. The term in the paper is mechanotransduction.
Right, mechanotransduction. What's fascinating here is that mechanotransduction is essentially exactly what it sounds like. It's the cell's mechanical to chemical translation system.
It's how it perceives the stiffness or softness of its environment.
Exactly. Now, for the longest time, the scientific community focused heavily on something called F-actin.
Which is basically the cell's muscle, right?
Precisely. If you picture the inside of a cell, F-actin acts like muscle fibers. It tenses up, it pulls, and it creates, physical tension.
Like flexing.
Right. But this new paper proves we've been missing a massive piece of the structural puzzle. F-actin isn't the whole story at all. We actually need to be looking at a completely different system.
Which is what?
The microtubules. If F-actin is the muscle pulling on things, the microtubules are the cell's internal scaffolding. They act as the structural support beams and, crucially, the cell's internal highway system.
The highway system. Okay, I want to pull on that thread. Because if the microtubules are the highways, does that mean the roads literally change their routes depending on whether the cell is squished or stretched?
They absolutely do. And the researchers managed to capture this visually, which is just stunning.
Oh, wow. How did they do that?
They used a technique called immunofluorescence imaging. Basically, by using special glowing antibodies, they can light up this invisible skeleton in 3 dimensions.
So they can actually watch the architecture change in real time.
Exactly. They tested cells in 2 completely distinct environments to see what the highways would do. They call these environments the mechano-on state and the mechano-off state.
Okay, let's paint a picture of that for the listener. Walk me through the mechano-on state first. This is when a cell is stretched out on a stiff surface.
Yeah. Imagine a cell sitting on a rigid surface. The researchers measured this at about 40 kilopascals.
40 kilopascals. What does that feel like?
It translates to a fairly stiff environment. Think of the consistency of stiff gelatin or certain dense tissues in your body, like bone or cartilage.
Okay, got it.
So in this stiff, stretched out mechano-on state, the glowing images show a massive, prominent central hub located right next to the nucleus.
Like a central station in a city.
Exactly like a downtown hub. Biologists call it the microtubule organize center, or the MTOC. It's marked by these specific proteins called gamma-tubulin and pericentrin.
Gamma-tubulin and pericentrin, so they build the station.
Right. And from this massive central hub, all of the microtubule highways shoot straight outwards, radiating toward the far outer edges of the cell, like the rays of a star.
Oh, the paper calls that an astral arrangement, right?
Yes, astral, like a star.
Yeah.
It's a highly centralized setup. Every single road leads directly back to the center.
But then they took that exact same cell and crammed it into a tiny confined space. Or they put it on a really soft, squishy surface, something like 0.7 kilopascals.
Right, which is more like the consistency of brain tissue.
Yeah.
Super soft.
And that's the mechano-OFF state. So what happens to the star shape then?
This is where the architecture essentially rebels. In that soft, squished mechano-OFF state, that entire central hub just vanishes.
Right, downtown is totally gone.
Completely dismantled. Instead, the inner ends of those microtubule highways detach, float away, and anchor themselves to randomly scattered points all around the outer edges of the cell.
Scattered points, like where?
They anchor to these complexes called gamma-TURCs at the cell periphery. So they lose that radiating star shape entirely.
And what do they look like instead?
The structural beams end up wrapping around the nucleus like a protective inverted cage. They call it an apicobasal cage.
It's basically redesigning a city transit system on the fly.
It really is.
Like in this stretched out, stiff state, you have this massive downtown hub, and all the train lines lead straight into the center of the city. But the second the city gets squished, downtown is obliterated, and the tracks are ripped up and relayed to just loop around the suburbs in this cage-like ring.
That is a brilliant way to visualize the geometry. As long as we remember, we aren't talking about literal steel tracks and trains, but, dynamic protein tubes that assemble and disassemble in seconds.
Right, it's super fast.
Yeah.
Okay, let's do a quick recap of this first part for the listener.
Good idea.
Point 1, cells feel stiffness, which is the mechano-OFF state, and they build a central hub with radiating tracks. Point 2, cells feel softness, the mechano-OFF state, and they dismantle that hub to build a protective cage around the nucleus instead.
Spot on.
So here's a question for you listening at home to think about. If you were a protein trying to commute to the nucleus, how would this architectural shift totally change your route?
It would be a nightmare commute, that's for sure.
Totally. Which logically brings us back to our executives. Now that we understand the shifting tracks, how does this actually control YAP and TAZ trying to get into the nucleus?
Well, if we connect this to the bigger picture, YAP and TAZ desperately want to enter the nucleus to turn on genes.
Genes like CTGF and CSERA61, according to the paper.
Exactly, those are the specific growth genes. But the researchers noticed something weird. In a squished, soft mechano-OFF cell, YAP and TAZ are mysteriously trapped out in the cytoplasm.
The cytoplasm being that jelly-like fluid outside the nucleus, right?
Right. They are physically locked out of headquarters.
And the tracks being rearranged is what locks them out.
Not directly. The rearranged tracks activate a gatekeeper. Enter a family of proteins called AMOT, specifically AOTP130, AMOTL1, and L2.
Okay, so AMOT. The paper describes AMOT as a cytoplasmic sink.
Yes, a sink.
Which is a bit abstract. I'm imagining AMOT more like a giant sticky sponge or, honestly, like a massive bouncer standing outside a club.
A bouncer is the perfect analogy.
So when AMOT is floating around in that cytoplasm, it physically grabs onto YAP and TAZ and just holds them in a bear hug so they can't move.
Yeah, it literally traps them. And the researchers didn't just hypothesize this, they proved it using one of my absolute favorite techniques called the proximity ligation assay, or PLA.
Break that down for us. How do you prove two microscopic proteins are hugging?
It's an incredibly elegant molecular trick. You take a glowing tag, split it directly in half, and attach one half to the YAP protein and the other half to the AMO bouncer. If YAP and AMOT are just floating near each other, nothing happens. But if, and only if, they physically touch and bind together, the two halves of the tag zip up and emit a brilliant fluorescent glow.
So it's a literal smoking gun. If you see the glow, they're making physical contact.
Precisely. And when they looked at these soft, squished mechano-OFF cells under the microscope, the cytoplasm was glowing brightly.
Oh, wow.
Yeah, AMOT and YAP were absolutely holding hands in the fluid outside the nucleus.
That is so cool. But how do we know AMOT is the only reason YAP can't get in? Like, what if the nucleus is just closed for business because the cell is squished?
Because they tested that too. To be absolutely certain AMOT was the definitive gatekeeper, they used CRISPR gene editing to literally delete the AMOT protein from the cell.
Just wiped it out.
Wiped it out completely, or they used something called SRNA to silence it. They took a squished, soft cell, deleted the bouncer, and suddenly YAP and TAZ flooded straight into the nucleus anyway.
Wait, really? They totally ignored the fact that the cell was squished.
Completely ignored it. Without AMT physically holding them back, they went right back to shouting growth orders. AMA is definitively the gatekeeper.
Wow. Okay, Dave, for another quick summary. Point 1. YAP and TAZ want into the nucleus to drive growth. Point 2. AMD is the bouncer that traps them in the cytoplasm. Point 3. This trapping only happens in the squishy mechano-OFF states.
Perfect summary.
So here is the next question for you to ponder. If AMD traps YAP in the soft state, where does the bouncer go when the cell is stretched out and actually wants to grow?
And here's where the transit system comes roaring back into the story.
Yes. Here's where it gets really interesting, because in those stiff mechano-ON cells, we know YAP and TZ are free. That means the AMT bouncer must be gone. How does the cell get rid of it?
It throws the bouncer in a transport vehicle and sends it to the shredder.
The shredder.
Seriously. Remember how in the stiff mechano-ON state, the microtubule tracks all point directly inward toward that massive central downtown hub?
The MTOC, right, the star shape.
Exactly. Well, the cell uses molecular motors, specifically a complex called dynin and dynactin. You can picture dynin as a microscopic machine that literally walks along the microtubule tracks.
It actually walks.
It has tiny little feet that step along the protein tube, pulling cargo behind it. And in this case, the cargo is our bouncer, AMO.
That is wild.
The dynin motor physically hauls the AMO protein along the highway, dragging it away from the edges and straight toward the center of the cell, dropping it off at that central hub.
Okay, so AMOT is relocated downtown, but that doesn't destroy it, right?
Not yet. But once AMO arrives at the hub, it gets tagged for the trash. The local enzymes in that pericentrosomal area slap a chemical tag on AMO through the process called perylation.
Perylation.
Yeah, by enzymes called TNKS1 and 2. Think of it as putting a sticky note on it that says, inspect this. Once that first sticky note is on, an E3 ligase called RLN-F146 reads it and attaches a long chain of molecules called polyubiquitin. Polyubiquitination is the universal cellular barcode for, destroy this immediately.
It's the biological kiss of death.
It really is. Once AMOT has that barcode, it is shoved into the pericentrosomal proteasome.
A proteasome?
Yeah, which is essentially the cell's local downtown recycling center. Or, to stick with your somewhat violent metaphors, a wood chipper.
Nice.
It shreds the AMO2 protein down into basic amino acids. And with the bouncer completely shredded, YAP and TAZ are finally free to walk into the nucleus and kickstart cell growth.
Okay, wait, I need to stop you there and push back on this, because the logic of this seems completely absurd from an energy perspective.
Oh, how so?
Let me get this straight. The cell is constantly manufacturing this AMOT bouncer protein, only to immediately load it onto a walking motor, ship it downtown, slap a barcode on it, and throw it into a wood chipper, just so YAP can stay active. That sounds like an exhausting, massive waste of energy. Why build a complex protein just to constantly shred it?
I love that you brought that up because it does sound super inefficient at first glance. It is highly energy consuming.
Right.
But you have to view it through the lens of survival. It is actually a perfectly elegant system for instant responsiveness.
Instant in what way?
Well, if you are a cell, you have no idea when your physical environment is going to change. Someone might bump into you, a tissue might tear, pressure might shift.
Sure.
If you waited until you were squished to start manufacturing a bouncer from scratch, it would take hours. The cell might inappropriately divide in the meantime and cause massive problems. By constantly making AMT, but also constantly destroying it on these microtubule highways, the cell is balanced on a razor's edge.
Oh, I see. It's like keeping your foot hovering over the brake pedal while you're driving.
Exactly, because think about what happens the literal millisecond that cell gets squished into a soft environment.
The tracks break.
Yes, those central microtubule tracks detach and break apart into that cage shape. The central downtown hub vanishes. Suddenly, the dynin motors have no tracks leading downtown.
So they can't reach the wood chipper.
Instantly, within seconds, the destruction of AMOT stops. The bouncer builds up in the cytoplasm, grabs the executives, and shuts down cell division. It acts as a mechanical rheostat, like a dimmer switch, providing a real-time, second-by-second response to physical force.
That is just mind-blowing. Let's recap this third part. Point 1, in stiff mechano-ON cells, dynin motors drag AMOT along microtubules to the center. Point 2, AMOT gets tagged for destruction by those enzymes. And point 3, AMOT is destroyed in the proteasome, which frees YAP and TAZ.
Nailed it.
So listener, what stands out to you? Is it the fact that the literal structural shape of a cell directly dictates whether a protein survives or dies?
It completely blurs the line between chemistry and architecture.
It absolutely does. But, we can't pretend this mechanical system operates in a total vacuum. Earlier in the paper, the researchers spent a lot of time talking about chemical signals, specifically something called the Hippo pathway. If the cell has this elegant mechanical track system, how do the chemical hormones floating around outside the cell interact with it? Like, can chemistry override the tracks?
This raises an important question, honestly. A cell isn't just listening to physical squishes. It is constantly bathed in a soup of chemical signals from the rest of your body.
Right.
The Hippo pathway is a very famous chemical signaling cascade that usually tells a cell to stop growing and behave itself. And what this paper reveals is a beautiful convergence of the two worlds.
How do they converge?
Well, the Hippo pathway utilizes a specific enzyme called LATS kinase. When this enzyme is activated by chemical signals from the outside, it chemically modifies our bouncer, AMOT.
By doing what?
It adds a phosphate group to it. It phosphorylates it.
Okay, so it phosphorylates AMOT. What does adding that phosphate actually do to the bouncer?
It acts like a suit of armor. When AMOT is wearing that phosphate shield, it physically cannot be tagged with the trash barcode.
Oh, wow. So even if the dynon motor drags it downtown to the wood chipper, it doesn't matter.
The wood chipper can't grab it. It's completely shielded from degradation. So the chemical signals from the Hippo pathway and the physical signals from the squishy mechano-OFF state are actually teaming up.
The architecture breaks the tracks, and the chemistry provides the armor.
Exactly. Together, they stabilize the AMOT bouncer and ensure YAP and TAZ are safely locked out of the nucleus.
Physical and chemical signals literally holding hands to put the brakes on cell growth. That's a beautiful system.
It is.
But whenever we talk about the brakes on cell growth, my mind immediately goes to the dark side of this, which is cancer.
Right, inevitably.
We know that YAP and TAZ are notorious in the oncology world. When they get out of control, they tell cells to proliferate endlessly, which is exactly how tumors form. So if this dual brake system is so perfect, how do cancer cells manage to hijack it?
Because cancer is, at its core, biology gone rogue. The researchers looked at specific cancer-causing mutations known as ROS and RTK oncogenes, and they found that these mutant genes corrupt this exact architectural checkpoint.
Do they corrupt the tracks or the bouncer?
They override the AMOT sink entirely. In a healthy cell, when it gets crowded by its neighbors, it feels that physical squish, stabilizes AMO, and politely stops growing. It respects its boundaries.
But the cancer cell doesn't.
No, these cancer mutations act like a wrench in the gears. They literally break the AMOT bouncer's ability to hold on to YAP and TAZ.
So even if the cell is incredibly squished, like tightly packed inside a growing tumor, the bouncer's just broken.
Exactly. The cancer cell ensures that YAP and TAZ are constantly flooding into the nucleus, driving explosive, unscheduled tumor growth, regardless of how crowded the environment gets.
Wow.
This paper effectively identifies AMO as a newly discovered tumor suppressor. Its very existence is a vital brake pedal on cancer.
Time for a final recap. Point 1, the Hippo pathway chemically shields AMO from destruction. Point 2, cancer-causing oncogenes bypass this system to keep YAP active. And point 3, AMO is acting as a newly discovered tumor suppressor.
A critical one, yeah.
Which brings us to our final listener question. If AMOT is the brakes on tumor growth, how could we theoretically help the cell hit the brakes?
That is the million-dollar question.
So what does this all mean? We started by talking about a single cell as just a microscopic bag of fluid. But what we're really looking at is a highly structured, dynamic architectural entity.
Absolutely.
The physical layout of the roads literally decides which proteins are shipped off to die and which survive to turn on our DNA. It's a mechanical continuum.
It is a mechanical continuum, and understanding that continuum leads to some thrilling implications for the future of medicine.
Like what?
Well, think about how we currently treat cancer. We usually try to drug the mutated genes themselves, or we use chemotherapy to just poison any rapidly dividing cell in the body, which causes terrible side effects.
Right, it's a scorched earth approach. It hurts everything.
But this paper opens up a radically new frontier. If AMAT is the tumor suppressor, and we now know it gets degraded by riding these dynon motors to the center of the cell, what if future therapies don't target the cancer's DNA at all? What if we target the transit system?
Oh, I see where you're going with this.
Yeah.
Could we design a drug that acts as a structural roadblock, something that literally cuts the tracks?
Precisely. What if we created a highly targeted drug that simply inhibits the dyning motors or gently dismantles that central downtown hub? You could artificially trick the cancer cell into thinking it's in a squished, soft state.
So even if the cancer cell wants to grow, it doesn't matter. You've cut the train tracks.
Exactly. The bouncer can't be dragged to the shredder. AMO builds up in the cytoplasm, grabs onto those hyperactive YAP and TAZ executives, and locks them out of the nucleus for good. We could potentially stop tumor growth not by attacking the tumor's genetics, but by rearranging its internal furniture.
Rearranging a cell's furniture to stop cancer.
Yeah.
That is profound.
It's a whole new way of looking at it.
It really is.
Yeah.
It all comes back to that initial thought, how does a cell feel? We've learned that it feels through tension, through architectural tracts, and through molecular motors rushing toward a central hub.
And it's happening constantly.
Yeah. The wild part is, this isn't just happening in a petri dish. If you're listening to this while out for a walk, every single time your foot hits the pavement, the cells in your bones and muscles are feeling that physical force.
They are actively tearing down and rebuilding these microscopic highways inside of you right now.
Just to keep you functioning. Thank you so much for joining us on this deep dive. Keep wondering about that microscopic universe inside of you, reacting to every single stretch and step you take.